I'd say let's go ahead and get started on our afternoon session here. So it is my great pleasure to introduce our second speaker and my longtime collaborator, Dr. Jake Gardner, who is visiting us from the University of Pennsylvania. Jake and I did our PhD in the same lab under Philly Weinberger at Cornell. And since then, Jake was also a postdoc at Cornell, a research scientist at Uber AI Labs, and now is an assistant professor in the Department of Computer Science at the University of Pennsylvania. Jake's research spans the spectrum of probabilistic machine learning, long-time focus on Bayesian optimization, and has in the last 5 years really been a leader in AI for science and scientific discovery. And he is going to give one of the coolest talks that I've seen in recent memory, not to hype things up too much, with that high bar for you, Jake. Please take it away.
Okay, I will do my best to live up to that.
No pressure.
Yeah, so great to be here. Always good to have an excuse to visit Vancouver for the 5th time in 2 years. Yeah, so our group, broadly speaking, works a lot on, I call it like structured design problems. And the goal here is I want to design some kind of object in some domain that has some properties that I want. So this might be antibodies that bind well to a target I care about. This might be materials. I actually don't know that much about materials. I have like a few collaborators that do, but like probably the domain I know by far the least about. At Penn, we happen to care a lot about RNA and nanoparticles. But even outside of chemistry, we also do things like database queries. So can I optimize the query plan that the order that I'll join tables in a SQL query in the database, right? And these are all really interesting problems. And the reason I'm putting them all on the same slide is because we have this sort of framework for doing this kind of thing that uses Bayesian optimization. And then that framework, I'm going to introduce it in a second. It's not going to be the focus of my talk today. My focus on my talk today will be talking about why I think all of that stuff is really bad, and why I think it could be way better. And when I say that stuff, I mean my own work. So I can call it really bad. That's okay. So actually, I probably shouldn't, because the student doing all this is on the postdoc market right now. So this is all amazing work.
Actually, going to be UBC in like an hour.
Like, oh boy. Okay, so we have been looking a lot at this idea that is not our idea originally, but I think it's a really good idea called late space Bayesian optimization. And the idea is I can train generative models in structured domains. So for example, let's say I'm trying to make antimicrobial peptides. These are small, I don't even want to call them proteins, but small sequences of amino acids that kill bacteria, right? Hopefully pathogenic bacteria specifically. And so I can train a generative model on these things because I can go get a huge database of peptides, right? I can, there's more protein, there's more unlabeled protein data on the internet than we could ever want. I mean, that's probably not true, but there's a lot of it. And I can train a generative model. And at a high level, what a generative model really does is under the hood, it has this thing called a latent space, which is sort of this is like a real valued vector space, right? It's just a pile of numbers that I can sort of move around in. And wherever I move in this space determines what peptides I'm going to get out, right? So the green point gives me the green peptide, the purple point gives me the purple peptide, orange the orange, blue the blue, right? So how does the generative model work? Well, a lot of times what it looks like is I'll do some kind of random sampling procedure in the latent space. With the VAE, that might be variational autoencoder, that might be sampled from a Gaussian, push through a generative model. It might be some kind of diffusion process where I also sample from a Gaussian and then push it through an even more complicated process to get a peptide out, right? Whatever that process is, there's some randomness that happens here, and out comes peptides. And the idea is, well, okay, let's use this to turn the structured optimization problem over peptides into a continuous optimization problem for latent space. Right, so what we lose here is I'm no longer directly optimizing over peptides, right? If I'm optimizing over the set of length 50 amino acid sequences, I'm not guaranteeing that I'm going to be able to discover every corner of that combinatorial space. But what I'm gaining here is I now have a real value to, instead of a discrete optimization problem, right? Like what amino acid do I change to what? I now have a continuous optimization problem, right? Because I can take that green point and I can perturb it by epsilon. right? And if I turn by a small enough epsilon, I probably get the same peptide out. But you know, like there exists some meaningful epsilon that like the smallest thing I can do to the green point get it like a slightly different peptide, right? And so, okay, how does this work with Bayesian optimization? Well, I have some procedure that scores these peptides that say, yes, these two peptides killed, you know, some of the bacteria that I'm interested in at interesting concentrations, and these ones never killed them, or you know, only at like ridiculously high concentrations did they actually kill those bacteria, right? I'm boiling that down to checks and crosses, but you know, you would go measure this as like how much of the peptide do I need in solution to kill the bacteria, right? And then here comes the Bayesian optimization, right? So I can now take a GP surrogate and I can say, well, okay, I don't really care about what's happening over here, but this looks an awful lot like a normal Bayesian optimization problem, right? Where I now have some points that I've tried, and I have some scores associated with those points. And so I can train a surrogate model on this data to say, what should I do next in this space, right? So, you know, here I might say, okay, I want to try whatever peptide comes out at this point before I want to try whatever peptide comes out at this point, because this is closer to the things that have been working so far. And now, do latent spaces organize this way? No, not naturally, but one of the papers that we've written sort of gets you good generative modeling performance while reorganizing the space so that you do have this property. Doesn't really matter how that's done at the moment. So I try this peptide, maybe it doesn't work and I'm sad, but at least I've learned something now, right? So I do the standard Bayesian optimization thing. I update my surrogate model with that new data, and maybe I propose something up here next, and maybe that does work, right? I sort of iterate this process, right? And then, you know, the point is like, from the perspective of, okay, we're doing Bayesian, the fact that I have this giant, huge model in the middle producing peptides doesn't matter anymore, right? Like, this has abstracted that concern away, and I can do standard Bayesian. And this works well both in simulation and you know, in reality. So we've gone as far as mouse modeling, where what we did was we took some extinct peptides. These are peptides from extinct mammals. The thought being is that if we base our peptides off of those, modern bacteria might not be ready for them, because like those peptides haven't existed in nature in hundreds of thousands of years, right? And so you take some extinct peptide and you make it a little better, and you know, now you're doing competitively or outperforming modern FDA-approved antibiotics in last resort, right? Fine. And again, you know, like the Twitter account adds to all these scientific papers, I will add in rats, right? Fine. I don't do human testing. I'm a computer scientist. Okay, so you know, and you know, there are also, of course, in silico benchmarks. So there's this I would call it relatively toy, but you know, it's a nice set of molecular design optimization benchmarks called Guacamole. And we can do the usual thing. It's like, oh, as I evaluate more candidate molecules, my score goes up more fast than the other people's, right? Like that's the thing you do in Bayesian, right? You say, okay, here's us, here's them. And you know, this is in 2022. We're not state of the art anymore. Here's a 2025 paper. You know, this pink curve that's doing like way better than everybody else across Bayesian, across reinforcement learning, across genetic algorithms. Here's another Bayesian paper, and like we're now down here, right? So like what we did to them, they did to us. And note that instead of being like 120,000 evaluations, we're now down to like 20,000 evaluations, right? So between 2022 and 2025, Bayesian has really genuinely gotten quite good for this, right? We're probably like an order of magnitude better, and then this was probably, you know, from here, like here was the state of the art existing for just within Bayesian, right? Like neither of these methods are actually even Bayesian methods. So Bayesian went from here to here, and from 2021 to 2022, and then 22 to 25, we go here. We're doing like really well with these problems. I believe that Bayesian is genuinely state of the art on basically every Guacamole benchmark test, like one of the Bayesian papers, right? So that's great, but none of what I just talked about is what I actually want to talk about today. What I want to talk about today is the following. So this ranolazine MPO test, where ranolazine is a drug that's used to treat chest pain. And what the task is basically make some modifications to ranolazine that we know how to score, right? In silico, like we just run a program and it'll give you a score between 0 and 1, right? So essentially you can kind of think of this as loosely fined analogs of ranolazine that like might also treat chest pain. So, okay, here's this amazing result, right? You know, according to the score that Guacamole is giving us, it achieved a score of nearly 0.95. And again, the best possible score on this task is 1, because we could manually construct a perfect solution to this problem, right? It's just that, you know, the optimizer doesn't know that perfect solution. Here is you know, the top scoring molecule found by this method. It's kind of a crazy thing. But more importantly, this exact molecule was studied in this particular PubMed paper, and you can go look this up if you want to. And it's known to be toxic. And in fact, it is so toxic that they weren't investigating its use in chest pain. They were investigating its use as a chemotherapy agent, right? Which is not a thing we give people for chest pain, right? And so, like, literally, not saying like, oh, the molecule produced by this method is similar to this. Is this exact molecule is produced by this method? And so let me take this further than just an anecdote, right? Like I've shown you one molecule. So let me make this more systematic. Here, I'm showing you, I'm predicting, okay, does the molecule have good ADME properties? So ADME is absorption, distribution, metabolism, excretion. So basically like bioavailability. If I put this molecule in you, like do you just excrete it out immediately, or is there some chance that you actually like absorb it into your body and it stays around for a little bit, right? Like that's the thing we want drugs to do, right? Molecules aren't useful as drugs if they just pass right through you, for the most part, right? There's like, like the mega dosing vitamin C thing, right? Like vitamin C, if you take too much of it, well all of it that you don't need just goes through you, right? It doesn't go anywhere in your body. So this is a predictor for ADME. I'll talk about how I made this predictor later in the talk. This is a predictor for toxicity. So toxicity here means, it means a couple things. It means mutagenicity. It means hERG inhibition. There's like a few properties that like, if it has this property, it's obviously not a very good drug. And here is, I want both things. So I want the classifier to say, so let me explain these plots. What I'm saying is, here is my classifier's probability that you have a good ADME profile. So I'm saying like at the x-axis 90, I'm saying my classifier says there's a 90% chance the ADME profile is good. The y-axis is measuring for all of these different molecular design methods. There's 11 methods here from the recent literature. What fraction of their top molecules did they get to keep? So we took the top 10% of molecules proposed by these methods, which are still millions of molecules, and we said basically what, reading this plot, what it says is, if you want at least a 90% chance of good ADME properties, according to this classifier, you only get to keep about 5% of your molecules. Okay? So of your top 10% of molecules, we're going to throw 95% of them out because we're filtering now. They're going to have bad ADME properties. And here, if I want 90% chance of both good ADME properties and no toxicity, none of these methods get to keep any of. By that point, essentially no method gets to keep any of their top molecules anymore. We filtered every single one of them out, right? So actually, every single one of these methods, what they return as their top method, as their top molecule, is the empty set, right? If you start filtering by these kind of properties. And again, I'm going to talk about how we built these classifiers in a second. Like in some sense, this is the point of this talk. Like how did we do this, right? And where I want to head with this is, sort of like the high level overview is, I think what we need to be able to do is say, okay, I want to optimize a new antibiotic. We know what that means, and we're pretty good at measuring that really quickly in the lab. Like, what does that mean? It's like I put the peptide, in a well, I put the bacteria in a well, and I say, is the bacteria growing, right? If it's not, well, that's indicative at least, right? So I want to optimize a new antibiotic, but I want to specify properties of that drug. Like I want to specify the mechanism of action of that drug. I specifically want a membrane depolarizer. That's how I want the peptide to kill the bacteria. And I'd really like it to not be liver toxic because that's just a bad thing for drugs to be, right? And when I say specify these things, what it's going to turn out, what I mean is I want to literally be able to specify these in English text. I want a scientist to write down in English, I do not want my drug to be liver toxic. And we can build a classifier from that. And I'm going to show you, so this is basically what we've been doing for the past 3 or 4 years. And this is kind of where I actually want to spend my time focusing today. And then I will put the things together at the end that show that like this is maybe working. So before I get to what we've been doing, I do want to point out that like we're not the first people to think of this. So there's this company called Future House. They've built a system called Paper QA2. You can train an order of tens of thousands of papers, and it will do retrieval augmented generation for you. What I mean by that is you can ask a question. It will do a big k-nearest neighbor search on LLM embeddings to say, okay, from the entire 30,000 papers, where is the most relevant stuff? Pull that out, give that to an LLM and say, answer the question using this text from the literature's context, right? That's essentially what RAG is in a nutshell. And then there's also this effort by Google. Basically what they did is they took Gemma, they took an LLM and they fine-tuned it on supervised data from TDC, this Therapeutic Data Commons, which has small supervised data sets of like drug-induced liver injury, for example. They have molecules they've labeled as, yeah, we know that this molecule has the potential to cause drug-induced liver injury, right? So LLM plus clinical data. There's just a lot more out there, and this is kind of where we're heading. Is like, what if we just spent piles of money to like cram the entire scientific literature into LLM? Right. That's kind of where we're going. PubMed has about 37 million scientific papers. Of those scientific papers, it turns out that Penn Libraries has a license to about 27 million of them. So we downloaded all of those. We are, unlike some companies, being genuinely careful about like, okay, when we use a paper, let's like, every paper we're using, we do have a license to. If you didn't want to do that, right, there are an awful lot of papers out there on there's a pirate head on the image, right? You can infer the sort of resource that Sci-Hub might be, right? But the point is there's like huge amounts of papers. And we know how to map, like, there's at least a first pass way to know, like, okay, do these papers mention proteins we care about, or genes we care about, or diseases we care about, or molecules we care about, right? You can go to a website like Uniprot, which just stores proteins and peptides. Then I can say, okay, let me go to this protein, and let me just download every single paper that I have a license to that mentions that protein. Or PubChem has, it indexes like 40 million papers, so even a bigger subset than this that we have access to. And you can go to any molecule and say, let me download the 1,000 papers that this resource believes mention those papers. And, okay, we can do that. And then there are even tagging tools that you can use. So there's a tool that the NIH currently still maintains called PubTator, where I can type in, okay, a tag chemical, like chemical levofloxacin, and it will say, okay, I know that LVFX here is referring to that chemical, right? And then I can map that back to say a smile string or a selfie string, like some canonical representation of that molecule, right? Here it is levofloxacin again. It's not highlighting amoxicillin, but it knows what amoxicillin is, and it will do some effort to solve, like, okay, people will alternatively refer to levofloxacin in the literature as levo or LVX or LFX or LEV or whatever you feel like naming it in your paper to save space because you have a paper constraint, you have a paper limit constraint that's like 2 hours from the deadline or whatever. Like, we've all been there. And this gets actually even funnier with some diseases. So here is a paper that throughout the rest of the paper refers to early onset neonatal sepsis as EOS, right? Because it's implied by the fact that you're reading this paper that they're talking about neonatal sepsis, right? And they refer to late onset neonatal sepsis as LOS, because the thing they need to distinguish for you as a human reading this paper is whether they're talking about early onset or versus late onset in that paper, like later on, right? So like EOS arguably is a terrible acronym for neonatal sepsis, which is the thing that's been tagged here, right? But you can solve that problem. We did basically spend an entire year and a half building our own tagger for this. So here are some other fun examples of problems that you run into, and most of this is engineering, right? So it turns out that if your molecule isn't lucky enough to have a name like levofloxacin, because it's not an insanely popular molecule, it's probably a really long IUPAC name where we like systematically write down, we count the longest carbon chain and we write down all the functional groups and blah, 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 right? So people don't like writing that in chemistry papers any more than you want to read it. And so what they do is they say, well, we note that molecule 8C and 8D were 4 to 6-fold more potent than 8A and 8B. And at the end of the paper, there's a table that's like, molecule 8A was the following thing. Molecule 8B was the following thing, right? You have different ways that people refer to proteins. It turns out that if you refer to, I don't remember which is which, but I think if you, an unspoken rule in biology is that if you use a gene name and you do capitalize the first letter, you are talking about the human version of the gene. And if you don't, you're talking about the mouse version of the gene. So that's just a fun thing to figure out, right? And then, you do also have people who will have a picture of a molecule and then they'll write down an IUPAC name. And then despite the fact that they're professional chemists, their IUPAC name for that molecule is wrong. Which is, so there's a lot of, the point is there's a lot of normalization to do. But you can do all of this, you can search the entire literature for everywhere in the literature that has ever mentioned a protein, everywhere in the literature that's ever mentioned a molecule, or a disease, or a gene, and you can build LLMs that can do this. So basically what you do is you start with a very large language model doing this kind of tagging, and then you fine tune a much smaller one so that you can run it without spending hundreds of thousands of dollars. Which you end up doing anyways. I'm going to point out, that this precision and recall, what we're measuring here is here's a paragraph where we know what the tag is. Like we know this molecule is mentioned in this paragraph. Can you identify that the molecule is mentioned in that paragraph, right? Like that's the classification task we're measuring precision we call for. And like 92% is actually an underestimate because like most of these are like, oh, we've identified a cell line, right? And like that won't make it through a smile string normalizer anyway. Like we're not gonna like turn a smile string, a cell line into a smile string and add that to our data set, right? So the precision is probably north of like 98% here. And then what you do is you run this over the entire literature and you say, okay, what do I have at the end? I have this giant database of entities. So entities are every molecule that I ever found mentioned in the literature and every protein I ever found mentioned in the literature. And I'm saying, okay, and you know, for each molecule, so here's histamine, I'm saying I'm going to pull out every single piece of text that I found across 20+ million scientific papers that mentioned histamine at all, right? Just everywhere in the entire literature that mentioned this, or everywhere in the literature that mentioned this protein, this H2M, right? And I'm just going to build this database of like entities, which are just proteins or molecules, and text, which is just like if it's been mentioned in some scientific paper somewhere, we're going to tag it that. So some of this is public now. So we did a little bit of extra normalization to say like, okay, we took like 3 million of those papers, which end up being, you know, about 1 million molecules, and like it's pretty close to 500,000 proteins now. And what we said is, we said, okay, if I see a piece of text that mentions histamine, use an LLM to not just use that text. We can't make the text public because the text is copyrighted. So what we say is like, okay, here is a paragraph that mentions a molecule. If you can, LLM, extract a generalizable objective fact about that molecule from that paragraph, which isn't perfect, but you know, you can't copyright the fact that gravity is a force that accelerates you at 9.8 blah blah, right? You can't. So the point is like this is now like you can go publicly download this sort of like sanitized version of this, although behind the scenes we have like 10 times as much data. It's just like we can't like, you know, we can't make that. We're trying to find creative ways to make this public, right? We don't want this to be a closed source thing, and it's just it's a legal thing basically. Like a lot of, especially with like these LLMs, a lot of this is really undecided, and we're just doing the pen libraries tell us to do, right? So behind the scenes, there are about 20 million of the 27 million papers that we have available. That we've already downloaded the PDFs. We've run OCR on them using pretty expensive OCR procedures if they're older papers. And let me show you how you can use this kind of data. Like how does this connect back to molecular design or Bayesian optimization? And what we're going to do is we're going to build a clip model, and we're going to build a clip model for constraining Bayesian optimization. So one thing you can do with Bayesian optimization is constrained optimization, where the constraint might also be a black box that just measures some property you care about. I'm going to do this from literature. So if you haven't seen clip models before, I want to introduce them really quickly because it's pretty cool. Basically, what I start with is I start with some text. So here in the orange text, I have some text that's talking about nitroglycerin histamine, and it's talking about a disease, cluster headache attacks, right? These are the things that they're tagged with. And on the right here, I have some more text that's talking about methicillin-resistant Staph aureus, and it's talking about clindamycin, right? Okay, so there's a disease and a molecule. And obviously, not all text mentions both a disease and a molecule. It's just, you know, there's just examples that I pulled out, right? And the idea is I first train a text encoder. You know, I use some, actually, I don't even train the encoder. I use some really big off-the-shelf text encoder. And what a text encoder does is it embeds the text, right? So I take the piece of text, I turn it into a vector, right? I have real valued entries in the vector. And, you know, the green point there is some large, like 3,000 dimensional vector. The orange point there is some large 3,000 dimensional vector. And I do this for more text, right? So you know, what's going to end up happening, we hope, and I'll show you how this happens in a second, is, you know, here is a new piece of blue text that also mentions nitroglycerin. Right, and so because both pieces of text mention nitroglycerin, I want them to encode to similar vectors. Here I'm doing that like literal Euclidean distance similarity. Really, you measure cosine similarity, but you know the point is I want similar text to embed to similar locations in text embedding. And then I also have this pink piece of text that mentions vancomycin and MRSA, and maybe that will embed similar. You know, it's just some location close to the text mentioning MRSA, which is a related disease, and you know, clindamycin and vancomycin are both antibiotics, right? So how does this happen? Well, I also embed molecules, and the key here is that I'm going to use a molecule encoder into the same size link space. So if these are 3,000 dimensional vectors for the text, I'm also going to extract 3,000 dimensional vectors for each of the molecules. So, histidine, histamine, nitroglycerin, clindamycin, vancomycin. And the idea is, okay, so I want, here is actually the supervision for training these encoders, right? The supervision is, I want the embedding for histamine to make it close to the text for the embeddings of the text that mentions histamine, right? So this is histamine, the molecule. This is text that mentions histamine, and I want those vectors to appear close together, right? So, why will these two appear close together? It's like, well, I have these two constraints. Nitroglycerin is mentioned in both pieces of text. So I have, this orange arrow here is encouraging these to be close together and these to be close together. There's gonna be some triangle inequality that kind of pushes these two close, like this and this can't happen without this in some way. But especially across it's a big space, 3,000 dimensions. Yeah, I know you can hide distance in a lot of ways, but there's a lot of data here, right? Like we have 2.5 trillion tokens of text that are going into the text encoder, right? It's like a truly insane amount of data. And then same thing, happens over here, right? So I say, okay, clindamycin will appear, vancomycin will appear close to the vancomycin text, clindamycin close to the clindamycin text. And here, this distance is more implicit because probably somewhere else in my text is a piece of text that mentions both clindamycin and vancomycin, right? This is I don't know, some commonly used antibiotics are blah, blah, blah for this infection, right? That probably appears somewhere in the 2.5 trillion tokens of text that we're putting into this. And so implicitly, these will be close together, even if we don't here have an example of a piece of text that has them close together, right? So you do this, hundreds of millions of times. And then, the key takeaway here is all we're doing is embedding molecules close to the text that talks about those molecules. The only labeling here that's happened is I need to tag the text with the molecule it's talking about or the diseases it's talking about, whatever entities I care about, right? And that we just did with LLMs, right? It takes a long time. I mean, I don't want to say just did that, right? Because there's enormous amount of engineering. But the point is at no point am I mentioning at no point am I supervising the model with clindamycin is an antibiotic, right? If what I'm going to show you is going to learn that, it has to be purely from the text that it's using. So how do we turn this is essentially an unsupervised model for all intents and purposes, right? The only supervision happening is this molecule appears in this piece of text. So how do we turn that into supervision? What we say is, okay, let's say I want to build a drug-induced liver injury classifier. What I say is, okay, I'm going to get 10 example paragraphs, and this can either be from an expert or an LLM if we trust the LLM, which we tend not to. But say an expert, I'm just going to say, okay, find me 10 passages that mention a molecule in the context of it causing drug-induced liver injury, and find me 10 passages in the context of it not causing drug-induced liver injury, or of molecules that don't cause drug-induced liver injury. Think of this as 10 positive text examples of yes, this text is describing a molecule and it's saying that it does cause drug-induced liver injury. This text is describing a molecule and it's saying that it doesn't cause drug-induced liver injury. That no evidence for that was found. It's probably a better way to say that. It's saying that it doesn't cause something is harder. And so you get text like this. You say, in repeated rat studies, blah blah blah, right? There's some inhibition of an important liver enzyme or whatever, right? Some cytochrome. Okay, fine. So these are your positive examples, and you have your negative examples. And then what we do is we take these and we embed them into using the text encoder that we had, right? So now this is obviously a very cartoon version of what we hope this looks like. But what we're hoping for is that in some notion, the positive examples describing molecules that cause liver injury cluster, and the negative examples also cluster. Why would they do that? Well, the model was trained on molecules being described causing liver injury, right? And so, those closeness constraints may be respected just by training on a giant pile of data, right? And there's evidence that this works, right? CLIP was originally the way that that OpenAI did zero-shot image recognition. If I, what is in this image, even if it doesn't have a notion of what the class is, right? It can just do that directly from text. So how do we use this as a classifier? Now comes along a molecule, and I want to know, does this, should I say that this molecule causes drug-induced liver injury? What I do is I encode the molecule, and I get this all into the same space, right? The text and the molecules all live in the same happy vector space together. And I say, okay, so this molecule here encodes closer to text that describes drug-induced liver injury than does to text that describes not causing drug-induced liver injury, right? So in this case, I'll classify this as a positive example, right? And the point is, like, at no point in this process did I ever actually explicitly write down a data set of molecules that cause drug-induced liver injury. And in fact, the way I built the classifier was I literally started with the English words drug-induced liver injury, right? And surfaced a classifier from that piece of text, right? So how does this work? So here is zero-shot drug prediction, property prediction. I'm showing you precision and recall in a bunch of tests. So BBB is, we're trying to predict, does this molecule cross the blood-brain barrier, right? And here's our precision and recall for doing that. And how do we measure this? It's like, well, it turns out that we do have a labeled data set of molecules that cross the blood-brain barrier. So for each of these tasks, we took that clip art I tried to show you and expunged every molecule that is in that test set and training set that we know causes blood-brain barrier. And then we use that entire data set as a test set, basically, right? So like of all the molecules that existed in TDC, that like here's a data set of 600 molecules that we know cross the blood-brain barrier. We just removed all the molecules and all the text that ever mentioned those molecules from the clip training. And we basically say, okay, we're going to try and predict whether those molecules cross the blood-brain barrier without the clip model having ever seen either those molecules or any text that mentions those molecules. And with zero shot, you do reasonably well in some of these tasks. You get north of like 80% precision. And I think more importantly in a drug discovery context, you get pretty high recall. And the claim here is I think recall matters more than precision because I really care about producing molecules that like say aren't mutagenic. I don't care if I miss a few molecules that aren't mutagenic that would also solve my problem, as long as I can find a few that aren't mutagenic that do solve my problem. Like if there's 10 molecules that solve my problem and aren't mutagenic, if I find like 3 of them, I'm pretty happy with that, right? It doesn't matter that I classified the remaining 7 erroneously, right? In a drug design context. Yeah.
What exactly do you mean by recall?
By recall, I mean, think of all the drugs that do cause, that are toxic. Like how many of those drugs did I actually identify correctly, right? So precision is, like the sort of like the accuracy question on the side of that. And then recall is the like, well, did I actually find all, like, what fraction of the molecules that cross the blood-brain barrier did I say cross the blood-brain barrier, right? That's the like, you could figure out the equation equivalent to that, right? So the point here is like 100, essentially 100% of the molecules that do cross the blood-brain barrier, we did identify as yes, those cross the blood-brain barrier, right? We recalled all of the molecules. And then the low precision means that there were molecules that we had some false positives, right? Fine. So we did this for BBB, for hERG inhibition, and mutagenicity and liver injury. These 3 things together formed our notion of toxicity as a surrogate, right? This is like, this will cause heart problems. Mutagenicity is not a good thing, and liver injury also. I think these 2 are pretty self-explanatory, probably. And then bioavailability is also part of it. We have the lower precision there. Of course, you can use labeled data if you have it. So one thing you can do is you can say, okay, I'm going to take the 200 molecules I have labels for on this task, and I will actually fine-tune my clip model with those labeled data and then test on the test set, right? And that does improve performance, for easier tasks like does this molecule cross the blood-brain barrier, we're now north of like 90% precision and recall on both, right? So we can make pretty good classifiers even with small amounts of labeled data now, right? Is sort of the key here, right? The clip model with no labeled data gets us in a ballpark such that fine-tuning that model with even a handful of labeled data gets us, pretty good classification for these things. So okay, finally at long last, here's a Bayesian optimization result. So what have we done? We took 4 of the guacamole benchmark tasks, and yes, these are still just guacamole. We haven't actually tested like, okay, does our cytotoxicity classifier match lab? We have in one example, but I can't talk about it yet because it's with a biotech startup, and I don't own anything there. But the point is, like, here are 4 guacamole benchmark tasks, and the gray bar in each of these is what you achieve if you run Bayes opt without any constraints. So this is essentially what you would have been doing in every guacamole paper ever, right? Like you just try to optimize it. What's the best score I can find according to the oracle, right? I have no knowledge of the fact that there are constraints. The blue bar is take this set of molecules produced by Bayes opt and no knowledge of the constraints, and post hoc filter those molecules with the constraint model, right? So I'm asking the question, now I'm going to throw away every molecule that your optimizer found if it doesn't have good, if it doesn't have good toxicity profile, for example. Like it's not liver, either we predict it's liver toxic or we predict it's mutagenic, right? And so, here, for example, this drops the score from like almost perfect, like 97% out of all, 0.97 out of 1, down to like 0.8, 0.82, right? Like a pretty large drop in performance. Obviously, the scale here is, this needs to be corrected for the paper, but that's just anyway. The more important point than like the visual inaccuracy of these bars is you can recover most of this performance by constraining Bayesian optimization with these constraint classifiers. So I'm saying I now have a classifier that predicts liver toxicity and mutagenicity, and I want to again optimize these, but now from scratch I want to constrain it so that I'm only going to produce molecules that pass these classifiers, right? And in many of these cases, you don't do exactly as well as the fully unconstrained task, but you certainly do much better than if you were completely oblivious to these problems. And yes, this does assume that these classifiers are working, but again, my argument is not that like these classifiers are perfect. My argument is that it's better to use something than to be completely oblivious to all these design considerations. Especially when things like liver toxicity, these are considerations that like 40% of clinical trials fail for these reasons, right? They fail for nothing to do with are they effective. They fail for like, oh, it's unsafe, or it has side effects, or it has, whatever it is. And then of course we do systematically measure our classifiers. So we're basically here, what we're doing is we're taking our CLIP model. This at the time was like a pretty small, like 15 million parameter CLIP model. We're comparing it to a 2 billion parameter, this TX Gemma model by Google. And in aggregate, we outperform the 2 billion parameter model. We do about as well as the 9 billion parameter model across, each of these actually gets unpacked. This is the 8 toxicity tasks that used AUROC as a metric. And then the metric that we're given, TDC tells us what metric to use, right? For each of these tasks, they tell us how to set up the training validation set. Here are the, for the 2 tasks for toxicity, that accuracy was the metric, here's what we did, right? There's like quite a lot of tasks in TDC, and for each one of those, we're trying to be as careful as we can to make sure that like none of the molecules that appear in the literature are in the test set for these data sets, right? So we, each one of those tasks involve a separate training procedure of the CLIP model, where I'm basically saying I'm going to pull out all the entire test set of that task from both the molecule side and the literature side of anything that mentioned that molecule. Obviously, if you care about predicting for molecules that you've seen in the literature, these numbers get very high very quickly, right? Because I can just retrieve directly, like, yes, that molecule is liver toxic. That was pointed out in this paper, right? And these numbers skyrocket. But the point is, can we predict this for new molecules? So I think this is kind of where I'm basically done. This is kind of what I want to build. And I think like there's this broader thing of like what I want to do is I want to run Bayes-off, but I want to describe things I care about in natural language, whether that's like here are constraints I want your molecule to satisfy, or even just like hey, I didn't like that molecule very much, right? Like that feels like feedback you ought to be able to give the optimizer. I think that there's more to do. So like CLIP models are great, but I don't think CLIP models are like the end all be all here, especially because like the way we did retrieval was sort of brute force. Like we basically processed the entire scientific literature with an LLM. That's really expensive to do. And so right now, what a lot of what we're working on is how we build better information extraction schemes to do that more efficiently. So for example, like what if we just built a RAG system over, instead of 30,000 papers, over 20 million papers, right? You can ask questions and we'll try to answer those questions based on 20 million scientific articles. And we even want to do, like I think there's a lot of low-hanging fruit in extracting a lot from the literature. So for example, we're pretty sure we can build pretty easily the like largest publicly available database of proton and carbon, right? Like why not, right? Like, chemists have a pretty standard form. You can almost regular expression these out of the papers, right? So like, why not build that database? I think it would be like at least an order of magnitude larger than anything that exists publicly. And then I think there's also a lot of tasks that we want to do in AI that like, if I want to do yield prediction for some chemical process, if I don't have the procedure that was followed somewhere in the model, like how on earth am I going to do yield prediction? Like that seems implausible, right? Because the procedure could be I threw both of the constituents, I threw both the reactants out the window and I bought the molecule I was looking to make from Sigma Aldrich, and look, I had 100% yield with that procedure, right? For loose definitions of yield. And then the point being is like, I think there's like, we can just get yields. Like if I want to build a big database of yields that maybe even bigger than like the open reaction, the ORD, right? I can maybe do that by just extracting the literature. There's also like all of those tasks I showed you in TDC. I can probably like 10x the size of every single one of the supervised data sets, right? Like the liver injury task. There's probably more molecules causing liver injury that are described in the literature than are in that data set, right? So let's just pull all of those out and be like, here is TDC++, right? For every single one of those data sets. Wow. Yeah, that's kind of it. I guess this is actually probably a better conclusion slide, but I'm happy to take some questions.
Thanks so much. Yeah, time for a couple questions. Yeah, what's up?
Okay. Sorry, I have some basic questions about the classifier that you trained.
Yeah.
Yes, encoder models in the same space. The molecules and the text are mapped to the same space. And actually, my question is about how do you constrain them to be close? Is it explicit in loss function, for example?
Yeah.
Something that happens at the result of some
So what you do is you take a text encoder off the shelf, just go download your favorite pre-trained text encoder. You take a pre-trained molecule encoder, and then to each of those you add an adapter so the dimensionalities match. And then you fine-tune the adapters with a loss function that says embedding for a molecule and embedding for a piece of text that mentions that molecule, like, minimize the squared norm between those two vectors, right? And sum that over all molecule common embedding pairs.
So the correspondence between the molecules and text is known?
Yes, so that was basically step one here, where
I
we.
thought it is unsupervised.
It is unsupervised in this. So we did it, right? Well, what we did is we took every molecule in PubChem, we went through every single paper in the literature, and we said, find me every paragraph that mentions that molecule, and then tag it and say, okay, here is a piece of text, here's a molecule that right, like I guess it's supervised in the sense that we just did the data set construction. It's like unsupervised in the sense that the supervision here was like a lot of engineering effort into an LLM to do the supervision for us, basically.
And actually, is your model a classifier or it is only an embedding learner?
Is it a classifier or an embedding learner? I'm not sure I understand.
Embedding learner.
Oh, an embedding learner. Oh, it is a, so this model as trained is an embedding learner. This procedure here, where I embed text that describes yes drug-induced liver injury and embed text that describes no drug-induced liver injury, turns the embedding learner into a classifier, right? So the original contrastive, yeah, sorry, go ahead.
Sorry, so two phases, one for learned entities and one for class.
Yes, although there's no like no parameters are tuned here, right? There's like when I embed a molecule, I do a pure distance lookup to these examples, and there's no like, and I'm not being cheeky and like secretly using a non-parameter. Like there's no notion of learning happening once the embedding model is created, right? At that point, just like raw distance computations in the embedding space.
Sorry, my last question about your computational resource for training this model.
Yeah.
What type of computation?
Training this version of the model is not like terribly expensive. The main thing that was expensive here was the fact that we had to train like a huge number of copies of it to account for like the different train and validation sets for each TDC task. We are now training models like on the order of like 2 to 5 billion parameters. Those take like $10,000 to train per model. So they get very expensive very quickly. I guess the blessing of having access to like a $30 million compute cluster at the moment. It's just like, you can, yeah, this research is really expensive. Like I think at the moment, if we were using public cloud resources, we'd be spending like $30,000 a day, basically. It's very, very expensive research. But that is part of why we are trying to make as much of this publicly available as possible, or like if someone just like, yo, can you give us give us all the papers that mention, but like hypothetically, if you ask me like, give me a list of DOIs and paragraph numbers that mention this set of molecules, like we can just give that to you. And then like presuming that you can go download the papers, you could then use the raw text of this. Like this is what we can release just like raw publicly. But the hope is that like we're not just like setting this money on fire. We want like if you do chemistry or materials or any kind of research where you think that you could like learn something from 20 million papers or just build classifiers without enough data to do it, right? Like you don't have supervised data for the property you want, right? Like we could also just train that flip model and give it to you, right? Like, you know, there's a question of how we get around the copyright issues here, but like, yeah, hopefully we're not setting the money on fire. We think we're not, yeah.
Really cool work. I was gonna ask, like, do you guys have you guys tested any like de novo like relations or like maybe like hypotheses to like plot new papers on? Like,
So we are doing that right now. So we have a chemistry collaborator, and ironically, like my knowledge basically ends in biochemistry. So there's like chemistry is already a little out of field for me, but they have these like bubble partwidth couplings, a type of chemical reaction, and they want to predict catalysts that improve the yields of specific kinds of those reactions. And so we are trying to do that right now. We are also trying to produce like, we have a collaborator, you know, we have collaborations that are doing this, but like the real science always takes like 10 times longer than the machine learning piece of the science, which makes sense because they do things that are like physical and all I do is light money on fire with the compute cluster. But you know, the point is like, yes, I want to do that, and that's a thing we're working on, but like those papers will probably not appear for like a while, assuming they didn't work. And I, you know, like does this work outside of walkable tests? I have no idea yet. I'm just kind of hoping that it does. that's like the honest answer to that question.
so like you said that you saw better improvement with like more parameters, or like where it was. Have you played around with like the size, like maybe with 10xing the number of parameters and improve performance, or is it plateau at this point?
I don't. I mean, I think we did study this to some extent, but like not enough. I think, like right now, what we're trying to do is, it's probably not obvious that you should be training the clip models on every single thing ever either, right? Like probably what we want to be doing is subsetting to like a targeted set of molecules that are relevant to your task and a targeted set of questions, right? So at the moment, we're like fully focused on information extraction. Like I have this like 40 terabytes of PDFs that turns into order of a few hundred gigabytes of like markdown files once you do OCR on the 150 mark and then OCR. And like, I want to get data that measures blah. Like, how do I get that exhaustive set of data out of the literature is a thing that just like retrieval can't do right now. Like you just go ask ChatGPT, list every molecule that crosses the blood-brain barrier. It'll probably give you like 5 or 10 or something, right? No matter how many it gives you, it's some small number that implies terrible recall, right? And so, you know, like should I even be building clip models, right? Maybe what I should be doing is just extracting a supervised data set of here's a molecule and here is text that describes evidence that it crosses the blood-brain barrier. Oh, this molecule was found in cerebrospinal fluid, right? Like that's not a clear-cut positive or one label you can do traditional supervised learning with, but it's a really targeted data set where I can now train like a sort of supervised classifier on, right? Because you know the classifier can learn to weigh the different kinds of evidence, like liver toxicity. You know, it inhibited this key liver enzyme in rats versus we did a population study of people taking this molecule and more people had liver injury than not, right? So it's significant. It's like, well, right, like they're just like different levels of evidence, and turning that into a classifier, I think, is at scale is just like an unanswered question of like how to do that. Whereas like when people are making clip models for image recognition, it's like, well, there's a dog in the image, or there's not, right? I think the way people give evidence for things in the scientific literature is much messier. And like, how do I weigh that population study against, you know, the liver enzyme study, right? Like both might be evidence, but they're not the same evidence. And so there's like a lot of questions for how to turn this correctly into a classifier. Clip was nice, because, so this paper was a data set paper. We were like, here's a data set. And the clip model was a way to be like, look, there's an off-the-shelf thing we can do to build a classifier right now and show that there's value by feeding some other people on these like supervised benchmarks. But like, how do we actually build classifiers from this information, I think is an unanswered question. Clip models do okay. There's probably better things, right? I hope there's better things. Otherwise, I'll just keep building clip models forever, but yeah. Yeah, sure, yeah, okay.
No, go ahead, yeah.
So I was gonna ask, when you were talking about the constraints, you said like for the Bayesian optimization, you had the constraints, so the gap wasn't as big. So like, what does that change in the Bayesian optimization side? Is it like too long of a story, or?
No, so essentially all you do, let's take the most general setting where both my objective function and my constraint are really expensive black boxes. What you do is you would design an acquisition function that that tries to trade off like satisfying the constraints and optimizing the objective. So a really natural thing you could do is measure expected improvement times the probability of the point is feasible, right? Where the probability of the point is feasible comes from your classifier and the expected improvement comes from your surrogate model of the objective, right? More recent, more sophisticated things, there's a paper, Scalable Constraint Bayesian Optimization, SCBO. They do this like Thompson sampling procedure where they like draw a Thompson sample, and then they basically like flatten out the Thompson sample wherever it's like it violates the constraints effectively, right? So that you're sort of like, I mean, you know, that there's like questions how to optimize such a Thompson sample, but if you're discretizing, it's fine. And then if you like don't yet have a feasible solution, you don't even try to optimize. You just try to like first find a feasible point, like just raw maximize probability feasibility, and then start doing optimization plus feasibility. But like the simplest thing that you could do that like works pretty well, if you, as long as you put like some of these high dimensional BO ideas on top of it, is just like compute EI and multiply it by like the probability that's feasible, or the probability that's feasible comes from your constraint classifier. Like, what's the probability that's liver toxic?
So is this some sort of like regularization type of thing, the first thing you described, or no?
I mean, it effectively ends up being regular. So basically what you're saying is like, I have an expected improvement. Here's how much, what is expected improvement is the expected utility of measuring this thing, right? You know, here's the data set that I have so far, the utility of that data set. Here's my expectation of the utility of the data set plus this extra point I'm collecting, right? That's what EI does. What I'm basically gonna say is, if I now measure expected utility, where I define utility to be that for an improvement of the objective, and the utility becomes 0 if the point isn't feasible. Like if you then work out that expected utility, you get just EI times probability of improvement, right? So think of it as like, okay, the utility is defined as my improvement if it's feasible and 0 if it's not feasible, right? Then just take the expectations, especially if you're like constraint and your objective, your modeling is independent because you have like a GP for the objective and a different model for the constraint, then like, that expectation splits apart and then you have probability of feasibility and EI. Fair.
What do you think are the limitations of these websites? And so you can make some very general classifiers, and I can imagine there's a lot of flexibility involved. So what do you always quote about it?
Yeah, I would say anything that's not like well studied in literature. So I'll give you an example of a problem that like a collaborator of mine wanted to work on that I'm like, we're gonna try it, but I'm not sure it's well suited for, is they're designing protacs, which are like, they're like actually quite large molecules that I put in your body, they go into your cells, they, one end attaches to some protein and the other end says, hey, come destroy this protein, cell the protein destruction mechanism. So it's a way to basically say, I want to destroy certain proteins in your body because you're making too much of it, you're making the wrong version of it, whatever it is, right? And the problem with protacs is that because they're so big, they have like really bad bioavailability. And the question is like, okay, can we make like a bioavailability classifier for protacs? And it's like, well, there's just like so few papers studying that phenomenon that like I'm not sure. I mean, we haven't tried it yet, but like I am skeptical that like this classifier is going to be accurate in that domain, right? Because it's just like it's like a pretty new chemistry, and it's not like a thing that's insanely well studied. Like a million people have studied antibiotics, right? And like mechanism of action, right? So I think like if you want to do like new chemistry to the extreme where like nobody's even describing that chemistry in the literature, this is like the danger of the clip model is, like you said, I can build a classifier for literally any property. If you can write down the property in English, I can give you a classifier for that property. And the question becomes like, how accurate is that classifier, right? And that's we restricted ourselves in this case to tasks where we had enough labeled data that we could like measure that. We could say how accurate is the clip model in this case, right? But like if you don't know, right? If you don't have any labeled data for your property, I can give you a classifier for that property. Like you just write it down in English and I'll give you a classifier. And then the problem is like, if that classifier isn't accurate, it's not more useful than like one that just doesn't exist, right? And so yeah, there's going to be a huge problem of like how do you determine the accuracy of a zero-shot model? Like by definition, that's hard because zero-shot implies you have no labeled data to measure performance on, right? So I don't know, you have to try it, right? Like you say, okay, you have to ask the question, like if I build molecules constrained by this classifier, do they have the property I want more often than if I didn't use it, right? And that's just gonna, you're just gonna have to pay the money to do that. Yeah. Yeah, I think coverage from literature is probably by far like the biggest thing that makes this like fail, if it's going to fail.
Right. And thank you for a nice presentation. And actually, I wonder where is your Bayesian optimization formulation? Is it in your objective function, which is sum of expectations, or is that right? Actually,
So like here.
actually, I'm looking for Bayesian optimization. Where is exactly the process?
Oh, so if I go like way back to the beginning of the talk.
From the viewpoint of implementation, I'm trying to imagine the implementation of your.
Oh, if you just want code, we have code like this.
No, just the process, yeah.
What does the implementation look like?
Yes.
So yeah, the simplest thing you can do if you're using like an off-the-shelf pre-trained generative model.
Yeah.
You take a generative model, and what does generative model do in a black box? It takes vectors of length 256 and turns them into peptides. So I now stick that 256 dimensional vector. I now measure f of generator of z, right? Is my objective function. So f is a function that measures antibiotic potency. Generator is a thing that turns z, which is a real vector, into a peptide, right? So I measure antibiotic potency of generator of z. And I just do optimization. I just treat it as a black box function in Z, right? So z is this 256 dimensional, 512 dimensional latent space in my generative model. And I just do, I just run the vanilla Bayes-Op turbo, whatever your favorite Bayes-Op is, or that, and then like the rest of this doesn't matter.
So is it experimental?
Where do the objective functions come from? Like where do the evaluations come from? It's a mix. So we have collaborators where we are directly doing optimization over a wet lab. And we also have collaborators that have machine learning surrogates of the properties they're looking for. And we do like multi-round, like we'll do some optimization in silico, go test in the wet lab, incorporate that feedback, do more optimization in silico, and like do those in sequence. And a lot of that comes down to like, the throughput of making some kind of things is much higher than the throughput of other things. Like RNA is like really expensive. It's like really low throughput compared to like lipid nanoparticles or peptides, right? Or like a question that's relevant there is like, is a grad student doing the pipetting or is a robot doing the pipetting? Because if a robot's doing the pipetting, we can probably optimize the lab directly. And if the grad student's doing the pipetting, we're probably not, right? Because the throughput's just like much lower because the grad student has to like sleep and eat.
I think with that, we'll call it there. Let's go to another round.
Wait for a second.